today's COVID-19 ECHO webinar, our first for 2022. As Mark said, back in April 2020, we didn't anticipate that we would still be here providing these webinars, but we still think that there's important and evolving information to share, and we'll continue as long as we feel these updates are necessary. So before we get going with this afternoon's program, I wanted to sort of take us back to December last year. Our webinar in December last year, we had to finish up quite rapidly because there was another event on, and I didn't get the opportunity to formally thank firstly the speakers from 2021, and all of them are shown in this graphic here. I'm not going to go through them one by one, but just to say thank you to all the speakers for really excellent presentations during the course of last year. And then Mark, if you can just go on to the next slide. And then, to say thank you to Project ECHO and Sanjeev Arora, who extended the license to us to use the platform. And as Mark said, everybody who's been involved in the administration of these webinars for the last 2 years behind the scenes. And finally, the last slide. Just to remind you that most of the webinars that we've held since 2020 can be viewed with a link shown there that is a link within our Department of Medicine website. So that's all for the slides, Mark. I think you can stop showing those. To move on to our program for this afternoon, we've got two important talks that really tackle COVID-19 from very different angles. One on the current epidemiology of the condition in the Western Cape, and then looking at immunological susceptibilities to severe COVID. We're going to take questions after each of the talks, a few questions, and hopefully there will be time for a general discussion at the end as well. So our first speaker is Dr. Hannah Hussey. Hannah is a public health registrar at the Western Cape Department of Health and at UCT. And Hannah's going to provide us an overview of the epidemiology of the Omicron-driven fourth wave in the Western Cape. So thanks very much, Hannah, and over to you.
Thanks very much, Graham, and good afternoon, everyone. I'm just sharing my screen. Can you see the presentation?
Yeah, we're seeing it. Yeah, and it's full screen mode. Yeah, thanks.
Great, thank you. So I'm going to be discussing the overview of the epidemiology of the Omicron-driven fourth wave in the Western Cape. And this presentation is going to have two parts. The first is the sort of more usual epi update. And then the second will be trying to tackle the question of the decreased disease severity that we've seen in this fourth wave. So to get straight into it, I'll start with the national trends. The Western Cape has exited the fourth wave formally last week Friday, along with all other provinces except for Limpopo and Mpumalanga. But slightly worryingly, however, is that even though the country as a whole has exited the fourth wave, we've now seen a slight uptick in case numbers in the last few days. And this is in several provinces, including Free State, Gauteng, Northwest, Limpopo, and Mpumalanga. We're still trying to understand this a little bit more. These cases are mostly in young people, and we're not sure if it's related to the return to schools and universities, as well as the loosening of lockdown restrictions. Related to this is the population level cycle threshold values. So the NHLS monitors the CT values on all public sector PCR tests in the country. And when the CT values drop, it indicates an increase in the population level viral load and can be an early indicator of a resurgence in cases. And as you can see, we've got a worrying drop in the CT values in Limpopo and Mpumalanga at the moment. Moving on to variants. So Omicron has been identified in 99% of samples sequenced in December and January in the country. There are now 4 sublineages of Omicron. BA.1 was initially the dominant sublineage, but BA.2 is increasingly being detected and now makes up around 23% of samples sequenced in the country. And what is still unclear at the moment is if the increase in cases that we're seeing in the northern parts of the country is related to an increase in BA.2, or whether it's just more related to the change in socialization patterns over the last few weeks. If you look at the Western Cape more closely, we can see that in mid-November, Omicron rapidly took over all the cases. But that since the last few weeks, we've seen an increase in the proportion of cases that are BA.2. This pattern is happening internationally as well. In some instances, it is associated with an increase in case numbers, and some cases, in some instances, it's not. And preliminary data from Denmark and the United Kingdom suggests that BA.2 might be more transmissible than BA.1. When we look at other differences between the two sublineages, so BA.1 is identifiable with S gene target failure, which is a proxy marker on PCR testing with a TAC-PAC assay, compared to BA.2, which lacks this proxy marker. So far, vaccines seem to be equally effective against both sublineages, and preliminary analyses from Denmark report that there's no significant difference in the risk of hospitalization with BA.2 compared to BA.1. However, there's still lots of uncertainty around the extent of cross-protection and whether infection with BA.1 will protect against infection with BA.2 is not clear. Coming back to the Western Cape and looking closer at our metrics, you can see that cases in the province peaked in late December. Since then, they came down sharply, and now we're starting, so case numbers are starting to flatten out again. We're still having around 500 new cases being diagnosed each day. Our test positivities come down to around 20%, and admissions and deaths have also decreased. So while we've definitely exited our fourth wave, we're still not at the low interwave levels we were previously. As you can see, in mid-November, we've seen much less COVID, only around 40 cases per day. And when we compare the cases in the fourth wave with the previous waves, we can see that the fourth wave, so that's the red line in the graphs here, had a much higher peak, but that the wave itself was narrower. So it started faster and ended faster than previous waves. And as Omicron is an immune escape variant, we saw a high number of reinfections. Places like Khayelitsha, which previously had a massive first wave, but then a very small second and third wave, again saw an increase of cases in the fourth wave. And similarly with the healthy working infections, despite widespread vaccination amongst these individuals, we saw a large spike of infections in the fourth wave. Moving on to the seroprevalence studies. So residual samples, which were taken for HbA1cs, were tested for SARS-CoV-2 antibodies. And you can see that overall there's a trend of increasing seroprevalence of around 20% after each wave. The Cape Town Metro is the black dashed line here. And so you can see after wave 1, we had 28% seropositivity, after wave 2, 47%. And in November 2021, after wave 3, we had 66% seropositivity. But there's heterogeneity in the sub-district experience of COVID, with the poorest sub-districts having persistently higher seroprevalence rates. And so when we look at this more closely, if we chart the seroprevalence against the percentage of low-income households in the Cape Town Metro, you can see that there's a clear linear relationship with the poorer communities having a higher seroprevalence. And while this doesn't necessarily sound like a bad thing, we just have to bear in mind that this immune protection comes at a massive cost. And so if we compare the seroprevalence to the age standardized COVID death rates in the first 3 waves, we can see that the seroprevalence was really, was only really attained through a massive loss of life. Moving on to the question of disease severity. So in previous waves, the cases, the admissions, and the deaths all tracked very closely with one another. But starting from the second half of the third wave, they started to decouple. And in the fourth wave, while we had this massive increase in case numbers, we didn't see the same increase in admissions and deaths. And so to unpack this a little bit more, Prof. Marianne Davies did an analysis comparing the outcomes of public sector cases in the start of the fourth wave compared to the start of the third wave. She did a survival analysis looking at the outcomes of admission, severe admission, or death. So those are the 3 boxes on the right. And then for each outcome, she looked at an analysis which only included age, sex, subdistrict, and comorbidities, and then a second model which included reinfection and vaccination status. And what you can see, sorry, is that in wave 4, wave 4 was associated with a lower hazard of severe outcomes when compared to wave 3, but that this effect is attenuated if you adjust for prior diagnosed infection and vaccination. You can see the hazard ratios are all moving to the right there. But the problem that we have in the Western Cape is that around only 1 in 15 COVID-19 cases are laboratory confirmed. And so we have a massive under-ascertainment of prior diagnosed infection. And this is particularly a problem when dealing with a variant like Omicron, which results in lots more reinfections than another variant such as Delta, which doesn't. So what she's done here is to additionally consider the effect of these unascertained infections. These are the purple triangles added to the forest plots. And here you can see that they attenuate the effect further, indicating that these unascertained reinfections are contributing to the decreased disease severity that we've seen in this 4th wave. But even after we take all of this into account, there appears to be some residual reduced risk of severe disease. And this is most likely the reduced virulence of the Omicron variant itself that we're seeing here. And then we did a comparison comparing the hazard ratios for the association between full vaccination and no vaccination for the same outcomes as before in both the September-October period as well as the November-December or the Omicron period. And here you can see in the later periods, the protection from vaccination was maintained. Unfortunately, when the same analysis was repeated trying to look at the effect of prior diagnosed infection on severe outcomes, there were so few individuals who had a prior documented infection as well as severe outcomes, that the confidence intervals here are too wide to be really meaningful. But comparing cases that occurred at different time periods can be challenging, and so to assess variant disease severity, ideally what you want to do is compare contemporaneous cases with whole genome sequencing. But this is obviously not feasible in most settings, and particularly in our settings. And so this is where the proxy markers really come into the fore. So Omicron, except of course for BA.2, results in S-stream target failure on the TACPath diagnostic PCR assay. And so using S-stream target failure data from the UK has shown that Omicron is associated with a lower hospital admission risk. And similarly, a large national study done by the NICD team led by Nicole Walton found that the cases with S-stream target failure had a lower odds ratio for admission of 0.2. Unfortunately, we do very limited TACPath testing in the Western Cape NHLS. We do, however, use more of the CG Allplex assay, which has its own proxy marker, RGRP targeted that can identify Delta specimens. And so using this proxy marker, we can then identify the non-Delta cases in the transition period of November to December, which are most likely Omicron. And you can see those non-Delta specimens that well with the S-stream target failure specimens, as well as what we have from sequencing as a whole. For this analysis, we included all public sector cases diagnosed on the CG Allplex assay from the 1st of November to the 14th of December. We used Cox regression for the outcome of hospital admission. Unfortunately, we couldn't look at severe outcomes like ICU admission and death because there were just too few of them occurring. And then we adjusted for age, comorbidity, vaccination status, and prior diagnosed infection. We included 1,600 cases with 122 admissions. And the main finding was that those without the proxy marker, so the suspected Omicron cases, they had a lower risk of hospitalization with an adjusted hazard ratio of 0.56. And then when we looked at the other results, vaccination was strongly protective with an adjusted hazard ratio of 0.45. And reinfection also seemed to be very protective. But unfortunately, we couldn't calculate a hazard ratio there because amongst those admitted, none of them had a documented reinfection, whereas amongst those not admitted, 12.5% had a reinfection. And then again, here we have the same problem where we can only really correct for laboratory confirmed reinfections. And this under-ascertainment of reinfection remains a major challenge when assessing the disease severity of Omicron. And the last thing I want to chat about briefly is the COVID-19 deaths in wave 4. So normally a COVID death is defined as a death within 28 days of a positive test, or during or shortly after a COVID-related hospital admission. In this fourth wave, we've seen a lower absolute number of deaths occurring. That's again the red line on the top graph. And similarly, when we look at the MRC excess deaths report, we can see that there's been a lower number of excess deaths in both the country and the Western Cape in this fourth wave. But anecdotally, clinicians have been reporting a change in the type of patients that they've seen in hospitals as well. And so to investigate this further, we did a rapid analysis of the first 50 deaths that occurred in the public sector admissions once we reached the threshold of more than 5 admissions per million population in the third wave, and we then compared this to the same group in the fourth wave. Two clinicians then independently used all available electronic data within the Provincial Health Data Centre to categorize these deaths. So the first category was those with COVID-19 disease, where we had evidence of COVID pneumonia. This was either the ICD-10 codes, the discharge of death summary, the prescription of steroids. And then COVID-associated, these individuals didn't have evidence of COVID pneumonia, and they had another medical condition that seemed to be more prominent. And it could be that the COVID just precipitated their acute admission here. So these included things like DKAs, strokes, as well as those with TB and cancer. Then, of course, there was an incidental group. So these were the ones that were clearly incidental. Either they were surgical patients or they were medical patients reported as having incidental COVID by the attending physician on the discharge or death summary. And then obviously there was an indeterminate category. Our main finding was that in wave 4, a much smaller proportion of deaths had evidence of COVID pneumonia compared to wave 3, and that this category of COVID associated also increased in wave 4 compared to wave 3. So what this does is it corroborates reports from clinicians as well as the oxygen consumption of facilities. So this graph on the top here illustrates the bulk oxygen consumption for the province with the green line being oxygen. And you can see clearly much lower levels of oxygen being used in this fourth wave compared to previous waves. And so while health authorities, including the Western Cape Department of Health, continue to report daily COVID-19 deaths, in the setting of increased endemicity, we need to be a little bit more cautious in how we interpret these statistics. So I'm almost done. Just to end off on my side, before the fourth wave started, we had a very high seroprevalence already in the province. When Omicron arrived, we saw very high case numbers in the fourth wave, including high numbers of reinfections and vaccine breakthrough infections, but a much lower morbidity and mortality was seen in this wave, caused by a combination of both increasing population immunity from vaccination and prior infection, as well as a decrease in variants with Omicron. We've also seen a rise in incidental admissions and deaths in this fourth wave. And so overall, the picture does seem to suggest that we are moving towards one of increasing SARS-CoV-2 endemicity, but just to bear in mind that endemic doesn't necessarily mean harmless, and neither does it mean that this is the end of COVID. And the increase of the BA.2 sublineage, the exact implications of that are not completely clear at this point, but that is a little bit concerning, as is the small uptick in cases nationally. And then just before I end off, some acknowledgements from my side to Professors Marianne Davies, Marlon Zhao, and Andrew Paul, as well as the team at the Provincial Health Data Centre, Western Cape NSLS, and the Effector Surveillance team. Thank you very much.
Thanks very much, Hannah, for a really excellent overview and bringing us up to date with a really lot of information that's been accumulated over the last 2 months, both in the Western Cape and nationally. So we've got a time for just a few questions. Hannah, if I can ask the first one, and that's, you showed us the data on immunity from prior infection, that had reached about 66% after wave 3 in the Cape Metropole. The data that, do you want to speak to the data that combines evidence of immunity acquired from both infection as well as vaccination, and what sort of levels we're at in the province?
Sure, we're still busy analyzing that data fully, so it wasn't quite ready to present. When we take, so the antibody levels that I presented earlier were the anti-N antibodies. So that looks really at this prior infection. In this last round of antibody testing, we also tested for anti-S antibody, and this is an indication of vaccine-derived immunity. And there, the levels were much higher, we're approaching 90% in most sub-districts. And so the indication is that between the combination of prior infection as well as vaccination means that there were very high levels of immunity in our communities prior to the start of the fourth wave.
Yeah. And are there plans for a further seroprevalence study? You know, given that it's likely that the remaining 10%, many of them were infected by Omicron in the last 2 months.
There's no plans at the moment. I mean, we can expect further increases in seropositivity moving forward, and what benefit that would do is not clear. Obviously, we're all tired of COVID work, and so the next we can do on it, but we'll see how things progress and what is needed at what point.
Okay, no, thanks. I just want to ask Mark if there are any questions in the chat.
Yeah, so there's one question from Tom Scriber, really looking at, is there any benefits between the J&J 1 versus 2 shots and the Pfizer vaccine in terms of breakthrough and protection?
So the formal epidemiological analysis of the vaccine breakthroughs in wave 4 is still pending and will come out shortly. In the data analysis that we've done so far and published so far, we haven't gone into that level of detail just yet.
Thanks.
And Hannah, if I can ask one other question, when you looked at the reproductive number, the R0 for the Western Cape, you know, it came down quite dramatically through January, but seems to be going up again and has reached 1. Does that, you know, is that an indicator that we're about to see resurgence in cases in the Western Cape, or what's the interpretation of that?
It's really quite difficult because there's lots of different things happening at the same time. The first is obviously that as we ended our fourth wave, it was obviously the festive period, and so there was decreased testing at that point, and that might have artificially increased or made the sort of decrease faster than what it really was. And now with the loosening of restrictions, the openings of schools, etc., some increase in cases is to be expected as well. So it's really difficult to say where we're heading. What the numbers do in the next few weeks.
Yeah. Okay, so there might be other questions, Hannah, that come up, and just encourage people, you know, to type them in the chat. And we can take them. So we're going to go on to the second talk for this afternoon, and that's going to be given by Professor Johnny Peter. Johnny is the head of the Division of Allergology and Clinical Immunology at Groote Schuur and at UCT. And Johnny's going to address a fascinating topic. We know that there are a number of very prominent comorbidities that predispose to severe COVID, but there's a subgroup of people, a minority, who, despite having none of these comorbidities, nonetheless develop severe and sometimes fatal COVID-19. And there've been obviously a lot of research focused on that internationally to identify genetic determinants in terms of those immune susceptibilities. And Johnny's going to summarize that research for us. Thanks very much.
Thanks very much, and good afternoon, everyone. And yes, I'm going to do my best in this talk actually to surprise everyone and try to use the word antibody and T cell as few times as possible. And there are a couple of concepts that I think that is worth talking about first. And this particular diagram, which is from one of my favorite, one of my heroes, my two married couple heroes for immunology from Yale, Kiko Awasaki and her husband Razlan Midhotov, who actually was accredited for discovering the toll-like receptors. And this is really the fact that the immune system works in a redundant way, but also in terms of an order of effective function engagement. And it really works from a lowest cost to a highest cost. And what's meant by that is a sort of that the immune system and activating different parts of it comes at different costs to host fitness. And that the order of engagement in this slide from sort of left to right here has an increasing cost. And really what we mean by that is kind of immunopathology and tissue damage caused by activation of the immune system. And I think this audience is very familiar now with, you know, one of our sort of main cornerstones of effective treatment for COVID has turned out to be, in a way, is immunosuppression or dampening down this immunopathology. And actually at the lowest level though, right here at the beginning on the left-hand side, are things that come with very low immunological cost. And these are largely constituently expressed factors of the immune system that occur at mucosal surfaces, antimicrobial peptides, secretory forms of IgA. And then slowly you go from left to right, activating first aspects of the innate immune system, and then later to things like antibodies and T cells. And as you kind of can't deal with an infection at one level, so you kind of go to the next level. And then the other concept, which has also been sort of fleshed out very nicely conceptually by these thinkers, is the concept of immune redundancy and its relationship to susceptibility to particular infectious pathogens. And you can see here in this figure, it's essentially showing that, you know, if you're a pathogen, let's say a virus, and you chopped up into various bits, you can be sensed by different aspects of the immune system, and that can lead to effector mechanisms which control the disease. And maybe in a particular individual, you have one particular effector pathway or sensing pathway that's defective, but as long as you have got an alternative, you will remain able to be protected from that pathogen. And this is sort of fleshed out down the bottom. And so there can be two pathogens and, you know, even the same pathogen in two different individuals that can be controlled in a way by two different aspects of the immune system. And on the surface of it, both of those people are effectively protected against the infecting organism. And then there can be some consequences, though, or some very interesting aspects of the host immune response that can be interrogated by particular pathogens. And so on the top here, and this may be something that we're seeing in COVID, which is that if, for instance, a particular person has a problem with one aspect of their immune sensing and effector mechanism, they need to control the infection with an extra effective or an alternative effective pathway may lead to that pathway being hyperactive or actually causing some immunopathology, like I was talking in the first slide. And then secondly, the alternative is that, you know, particular infections might have certain pathways of the immune system that are really non-redundant, and that only on exposure to that particular infection with its particular parameters or structures to invade host immunity are you actually able to see these specific defects in host immune system come to the fore. And that's really what I'm going to talk about today with COVID. And then just one general comment sort of from the field of inborn errors of immunity. It's really that the last like 20 years, but probably the last 40 years has seen a massive increase in understanding of genetic abnormalities of the immune system. And largely we've moved from this picture here, which is of one of the first cases of an X-linked common gamma chain deficiency, so-called, he was the bubble boy, so the first sort of well-documented case of severe combined immune deficiency. And we've now moved to this condition time where we've got over 400 single gene inborn errors of immunity. And what's interesting is there might only be a few described in a particular family, but collectively these conditions are now becoming increasingly common. And then the other aspect of inborn errors of immunity, which has received a lot of increasing attention, is the recognition that with these inborn errors, you might actually not only present with infectious susceptibilities or, you know, immune deficiency, as we previously know, but a lot of immune dysregulation. So simultaneously, a person with a gene problem in the immune system can present with a susceptibility to particular pathogen, as well as, say, autoimmune cytopenias or hematological cancers. And increasingly, we're seeing this balance. And so the first place in relationship now to SARS-CoV infection was that the primary immunodeficiency clinician community was very worried at what would happen when SARS-CoV entered patients with known inborn errors of immunity, the majority of which are children, so quite a young population. And what's been very interesting is there's now been several cohorts internationally that have been described. And this particular paper that I'm putting up is of 75 patients, aged largely up to their early 20s, with different inborn areas of immunity. You can see it's listed here in column 3, and it sort of expand from antibody deficiencies to hyper IgE syndrome, across to particular defects in neutrophil functions, or even T cell defects. And the remarkable thing, if you look here from the WHO ordinal scale, is the severity of their SARS-CoV infections was remarkably mild. Many were asymptomatic. And in fact, I think relevant as well to the later part of this talk is the 2 patients with a defect in MyD88 signaling, which is downstream of toll-like receptors. They were the 2 patients that actually presented with the more severe disease, even though it actually wasn't too bad. And then relevant as well, and this community will probably be more familiar with some of that research, is we have a lot of familiarity and experience, and we're very worried about how patients with acquired immune deficiency, particularly HIV, how they would fare, or patients that are receiving, for instance, B-cell depletion therapy with rituximab or other biologic therapies. And there's now quite a lot of data, and there's quite a lot of sort of registry data to really show that, for instance, HIV, the odds ratios for severe disease are somewhere between 2 and 3. So not really what we were expecting, potentially a 20 to 40-fold increased risk. It seems that rituximab, when you control for other independent risk factors and other comorbidities, particularly age, does not seem to increase severity of disease. So that's actually, essentially people that have largely had lots of their antibody production knocked completely out. And then also, if you look across biologics like anti-TNFs, you look in allergy, we look at IL-4, IL-13 blockers. There's actually been, there's a large study of over 7,000 patients that showed no real impact on death. And in fact, in those cohorts, time and time again, the biggest risk factors that seem to come out are age and male gender. So now moving attack from kind of the bigger group of inborn errors of immunity to really narrowing it down to 2 particular interesting considerations, which have really formed the focus and kind of the pioneer of this field, has been someone called Jean-Laurent Casanova from the Necker. He's now based at the Rockefeller Institute. And actually, with response to COVID, a lot of the work I'm going to show you is part of a consortium that now is over 400 investigators across the world that forms the Human COVID Genetic Effort. That was formed by Casanova and his colleague Helen Su, and also Jean-René Abel. And really the reason that they formed this was because of their background and their interest in the lab, looking specifically at susceptibility to particular pathogens. And probably the best way of conceptualizing the 3 things that I've put on here, the 3 columns, which talking about sort of the bigger group of primary immunodeficiencies, then Mendelian susceptibility to particular pathogens, and then the kind of concept of monogenic infections, is really to look at close to home, to look at TB, the example of TB. So, patients we see commonly every day with increased risk of TB with acquired immunodeficiencies. And if you look across sort of severe combined immune deficiencies, these are the patients that present with severe disseminated BCG or BCGosis. These patients are often in children, their gene defects have high penetrance, and they have a lot of infections across different types of pathogens. And then you get sort of specific defects in the immune system and what we call Mendelian susceptibility to infections. And a good example here is all the Mendelian susceptibilities that describe to mycobacterial diseases. And these are particularly gene defects in the IL-12 interferon gamma pathways. And what these patients present with largely is disseminated forms of non-tuberculous mycobacterium. And we actually, together with Jean-Laurent's group, described one of the first cases of interferon gamma receptor problems in a young patient that presented recurrently to Kreutzkia and Tigerberg with disseminated NTMs. And then more recently has been the concept of a sort of gene problems with low penetrance that cause sporadically particular susceptibility to infections. And Jean-Laurent's group probably is best known for describing defects of toll-like receptor 3 in cases of herpes simplex and encephalitis. And recently in TB, they actually described an area of homozygosity in a TYK2 gene that was enriched in European populations. And they estimate now that across TB, this accounts for about 1% of cases that develop active TB. So now moving on to explain a little bit of the early immune response that's critical to these gene defects in response to SARS-CoV. So we know SARS-CoV binds to the ACE2 receptor and its TMPS co-receptor, and then really it activates these toll-like receptor pathways, and particularly the toll-like receptor 7 and 3, which are the endosomal toll-like receptors that are activated by viral RNA. Downstream of these, their signaling pathway for the toll-like receptor goes through MID88 and upregulates NF-kappa B and pro-inflammatory cytokines. And then additionally, they also activate these, what they call interferon response elements or response factors. And particularly important here are IRF3 and IRF7. And then this leads to the production of type 1 and type 3 interferons, particularly interferon alpha and beta. These bind to their cognate receptors, and they then downstream signaling that involve largely heterodimers of STAT1 and STAT2. Those then activate interferon-stimulated genes, and they lead to the production of a host of inflammatory genes critical to activating the innate immune system and also, as well, the adaptive immune system. What's relevant to SARS-CoV on this picture and what appears in red and green are in fact proteins of the SARS-CoV virus that directly inhibit parts of this pathway. And so what that means is one of the main host invasive mechanisms of SARS-CoV is to inhibit the early interferon antiviral response. And this is possibly critical to part of the reason why SARS-CoV-2 has been such an effective and lethal viral infection. So on the basis of actually data from severe cases of influenza, where toll-like receptor 3 and IRF7 problems have been previously looked at, this group of the COVID human genetic effort went looking in about 700 patients with severe COVID across the ages. And what they found, they went looking specifically in these pathways. So they focused on these toll-like receptor and IRF7 pathways and related genes. And they went looking for particularly autosomal recessive or other variants that have functional significance. And what they found here was they found 8 deleterious mutations in 13 genes. And predominantly, they are highlighted here in this pathway, and it accounts for about 3.5% of patients with severe infections. What was good about this, and this is something that we face commonly now with the way genetics has come into primary immunodeficiency care, is you face a situation often where variants are identified and the functional significance of this in the immune system is unknown. And so really the work of this consortium has not just been in the identification of these genomic problems, but is in confirming that these variants are pathological and that actually impact the functional production of the immune system. So here is some lovely functional data which has been focusing on plasmacytoid dendritic cells, not dendritic cells, apologies for the typo there. And this is probably my favorite viral cell or immune cell fighting SARS-CoV-2 is the plasmacytoid dendritic cell. And here you can see this is on the y-axis is here production of interferon alpha and interferon delta there. And you can see here, this is in stimulation with influenza A virus and SARS-CoV viruses. And you see quite nicely illustrated here that actually SARS-CoV does inhibit, even in the control population, the production of interferons by plasmacytoid dendritic cells. But importantly, in patients with this IRF7 variant, P1, you see complete absence almost of these responses. So it really illustrates that this gene defect correlates with functional significance. And then further on, they've now looked at an expanded cohort of more than 3,000 patients, and they found other defects also in the interferon alpha receptors and downstream here in IRF3 and also in TRIF and TBK1. But perhaps another great set of science that's been done has been in studying the cohort of 1,200 unrelated males to particularly look to see whether there was any kind of hemizygosity or homozygosity in X-linked recessive disorders that could be found. And one of the target genes there was this toll-like receptor 7, which is a gene expressed on the X chromosome. And what they did is they went looking in this cohort for in and out of frame non-synonymous variants in the protein coding regions with very rare minor allele frequencies. And in this group, they found 16 unrelated male patients with critical COVID, 12 of whom had this loss of function mutation, and 3 had a hypermorphic mutation in toll-like receptor 7. And just to highlight, and I put this slide in just to show you the sort of extent of work involved in trying to find this. These slides show NF-kappa B activity, and they expressed as a percentage of wild type. And these are all a cell line that has had plasmid transfection of these particular variants put in to see whether they are functionally significant. And to find these particular mutations, they screened more than 100 variants that came up in their initial genetic analysis. So a huge amount of work put in here. Then related to this, and I'll change tacks now, and I'll just introduce one very interesting immune deficiency that is probably well known best to endocrinologists. And this is the condition known as autoimmune polyendocrine syndrome, or its full name relates to polyendocrine candidiasis and ectodermal dysplasia. And this was a condition, the clinical phenotype of which was known before the gene defect in the autoimmune regulator, or the AIR gene, which is critical in thymic development of central tolerance, was identified. And what's fascinating about this condition and its relevance to this is that the majority of the pathology in this condition relates to the production of autoantibodies. And so, for instance, if you look at the susceptibility to fungal skin disease, chronic mucocutaneous candidiasis, the defect here is anti-IL-17A and F autoantibodies. So a lot of these things, and this sort of these patients and their susceptibility to infections has kind of given rise to an interest in looking for something called phenocopies of primary immunodeficiency. So these are autoantibodies which relate and generate the same clinical phenotype as if you have a genetic immune deficiency. And probably known to this community potentially is autoantibodies to interferon gamma, which has been described to underpin some patients with mycobacterial susceptibility. And in fact, Stan Rest described a cohort as well from Groot-Schuur that had 2 patients with these autoantibodies. But relevant to COVID, they got really a great surprise and found 10% of this cohort that I mentioned of about 900 patients, and they've actually gone on to look in larger cohorts and see potentially up to 20% of all patients with SARS-CoV produce or have autoantibodies that can suppress the physiological levels of interferons. And so this essentially produces the same phenotype that I was mentioning. There's been a lot of effort around this and a lot of question about which came first. Were these autoantibodies there all the time? Have they been in fact generated by a viral infection? And so the first part of the kind of science of this was to look at the functionality of these autoantibodies that were detected. And these diagrams show this very nicely. So in the y-axis here, you see this is phosphorylation of STAT1, which is downstream of the interferon alpha receptor. And you can see here about patients that produce these autoantibodies, which are usually of the IgG isotype. They have a functional defect in interferon signaling. And you can see that they produce actually a wide variety of anti-interferon antibodies across sort of all the different isoforms of interferon alpha and also interferon omega. And much more rarely, there are some patients that are producing autoantibodies against interferon beta. And this picture here looks at the downstream genes, which are CLX10, which is IP10, and that's downstream of interferon stimulation. And you can see here that the autoantibodies are really demonstrating the functionality of these. And this control here just shows you that in these patients with autoimmune polyendocrine syndrome, they in fact are present already. And what's quite remarkable about this, if you consider that the odds ratio for diseases like diabetes, hypertension, HIV are sort of sitting in the region of 2 to 10 times, and probably age is our biggest predictor that increases odds for severe COVID at about 10-fold. These odds ratios are sitting in the 50s, 60s, so incredibly predictive of severe COVID. And the one question was whether or not these antibodies are there all the time or whether they come and arise from COVID. And probably the best data for this is in this paper here, which is a cohort of 22 of these autoimmune polyendocrine syndrome patients that got COVID. Remarkably, this is a young cohort of patients, and of the 22 of them, 19 required hospitalization, 15 developed pneumonia, and 4 died. So an incredibly severe course that these patients had. You can see from this diagram on the left that they all had autoantibodies pre-existing to different interferons, that these antibodies were functional. And you can see this is they're only at sort of 10 to the 4 dilutions of their plasma do you start to see a return of interferon activity. And then interestingly here, in one patient that they knew about this already from, they treated the patient with plasma exchange, and this actually could reduce autoantibodies in the plasma and make a big difference. So what about moving now out to the general population? Because the real question clinically is obviously, how common is this? And so they've now looked at it in a bigger cohort, about 34,000 patients. And it seems that these autoantibodies occur in about 0.53% of patients. But remarkably, there's a 10-fold increase in patients over the age of 70. So you can see it goes from this background rate of 0.5 to more than 4% in the older populations. And similarly, if you look in the critical cohort, critical COVID group, it's more than 20% in the over 80s. So it might, and this is what I mentioned that first slide, it potentially accounts for about 20% of patients with severe COVID. And kind of putting this all together, you can see here that, you know, as the infection fatality rate increases with age, probably the role of genetic problems is largely restricted to patients with children that develop severe COVID, but these autoantibodies and their role probably increase and account for more than their fair share of patients in the older population. And that relates here to this kind of determinism of critical COVID at different ages. And you can see here in children, probably these inborn errors might be as high as 15–20% of accounting for all children with very severe disease. In the adult population, there's still a lot of unanswered questions in terms of what roles different systems, including the immune system, but other things like the kallikrein system or the renin-angiotensin system dysregulation play in developing severe disease. And in the elderly, probably again, this is now accounting for 20% of severe disease. And really, it fits with this two-stage kind of pathogenesis model of critical COVID, the first being this early antiviral response, and then the second being the other higher order effective immune responses. And then what about clinical implications? Well, there are some of these papers that are saying that we should now be testing individuals for at least 3 of these interferons, and potentially more so in the higher priority groups or the older groups, or also in the autoimmune populations that we already have. We've also joined now and become part of this human COVID-19 genetic effort. And actually, we are working now on a shipment related to our own COVID cohort to look for these autoantibodies. And I invite, and this is an invitation to colleagues on this call that are potential collaborators that have cohorts that if you think this might be interested, please reach out to me. We will probably ship to them in the next month or 2. We've investigated setting up these autoantibodies in our own lab, but the colleagues in France and Rockefeller are being a bit proprietary about giving us the sort of recipes to set this up. And that might be relating to the fact that they're trying to commercialize these assays down the line, but we'll keep trying. And also if other colleagues have advice to help us setting up, please feel free to reach out to me. This will be very interesting. Thank you very much.
Thanks, Johnny. I'm just switching my camera on. Thanks for a really excellent overview, fascinating topic. So Johnny, the issue of the type 1 interferon antibodies, what are there other infections that are predisposed to severe disease as a result of patients exhibiting those?
Yeah, that's been the remarkable thing, is that it doesn't appear that way. So in all of these patients with the autoantibodies, they don't even have a history of severe influenza. And I think the argument that's put forward for this is what I showed in that slide about how SARS-CoV and other coronaviruses directly target these different aspects of the signaling pathway as their main host evasion strategy. So it's possible that, you know, other viral infections are not doing that to the same extent, meaning that, you know, if you have autoantibodies that only say inhibit quite low physiological levels, you still get away with it. And it's only when you have this like quite profound infection-driven suppression of these senses that you then have problems.
Yeah, and I mean, has the disease phenotype been well described in these patients? I mean, obviously they have worse outcomes, but you know, is it that they have higher viral loads? Is it that they develop more severe pneumonias? Is it more the kind of secondary immunopathology that's more severe? Has that been characterized?
You know, I think that there was some details in one or two of the papers that I've seen, but in the initial work and also the laboratory work has focused largely on the kind of functional defects and just with the like a binary outcome largely of critical ICU related COVID pneumonia. So that was always the starting point. I would have to go and there might be some of in the sort of supplementary materials of a lot of this work, there might be some of those details related to sort of IL-6 levels, CRP levels, that kind of thing.
And then just a final question from my side. I mean, I was under the impression that these inborn errors of metabolism were contributed to young people who developed severe COVID, but from what you're saying is that even in the elderly population, this seems to be a prominent factor in differentiating those who are going to get more severe disease than other people in the same age cohort.
Yeah, I think that's what's been surprising. And I mean, you know, as an adult immunologist, I've always been interested in kind of adult related immune deficiencies or problems related to the immune system. And I think these autoantibody conditions, and I mentioned that one about mycobacterial susceptibility, that was seen in an adult population. And I think this example with SARS-CoV is really kind of, in a way, turning, you've got the genetic defects, which predominantly are restricted, like that second last slide I showed, to probably severe children with severe disease. And then probably a much more important and bigger contributor to the burden of overall severe disease is these autoantibodies in this older population. Yeah. And in fact, people are now, the next phase that they're looking at is whether or not these are the group of patients that we're seeing with vaccine failure. So patients that develop severe disease after vaccination.
Yeah. Thanks, Johnny. So open for questions. Mark or Wendy, are there questions in the chat?
Graham, there's a question, or at least Ian Ross has raised his hand. Ian, I'm going to unmute you if you'd like to ask it.
Be great to hear from Ian about these patients with APS.
Thanks, Johnny. Thanks for a great talk. We have done some analysis of a worldwide Addison's group, and you know, as the vast majority happen to be autoimmune. And interestingly, consistent with the data that you've shown, there's been approximately 2 times or 2-fold risk of contracting COVID. And when we compared it to the background population, there was a 28-fold risk of hospitalization, implying significant COVID. So I think there's something, obviously it's not entirely and exclusively APS1, but you know, one would see a vast array of Addison's disease, but you know, one can presume that the vast majority are in fact autoimmune. So hopefully that gives us additional credence to something that we've seen and something I can relate to as potentially affecting the outcome amongst these people.
Yeah, and you should look for, you should follow that on by looking for these autoantibodies. I think that would be a useful, you know, mechanistic proof.
So I guess in a way it's a proof of concept.
Yeah.
Mark, no further questions in the chat.
No, nothing at the moment.
I mean, Johnny, just looking to the future in this research field, do you think there's likelihood that there will be, you know, further discoveries of important and common genetic abnormalities that predispose to severe COVID, or do you think given that the vast sample sets that have already been analyzed, that you would only now discover less common gene deficits?
Yeah, I mean, I think I would be, there's still this ongoing effort, and we, like those populations, I mean, one of the reasons we were looking to join this consortium was because there largely is underrepresentation of African genetic backgrounds and populations. So that's something, and also these autoantibodies, I mean, we don't know. And so that would be useful to expand to other populations. But, with 400 scientists, they do have pretty good global coverage. They've got most of Asia, a lot of Asian populations. They've got European and American populations. And South American populations as well. So it's quite widely distributed across the world. So I'd be quite surprised if now we find, things with such large odds ratios and effect sizes. Probably what we'll see now, and this probably does explain a larger set of severe COVID, is that it's probably a heterogeneous group. So, and maybe what I was saying right at the very beginning about how, one of the consequences where somebody might have some a kind of a variant in their immune system, which then allows these other effector pathways to be overly dominant. There could also be, and I suspect this will happen, there might be smaller effect sizes for genes in other systems. Like, we particularly interested in the kallikrein-kinin system and the RAS system and how that relates because of our angioedema work. And so probably we'll find smaller effect sizes to explain the larger group. That also might be different from population. So, in terms of like a single test, I think maybe these autoantibodies definitely might hold sway as having like quick and immediate clinical application.
And have there been, just picking up on that point about the RAS kallikrein system, have there been gene deficits demonstrated in those systems that associate?
Not yet. I think that we're working on the sort of physiological side of that, which is quite difficult. And again, I've not seen any publications yet about specific genes in that pathway being problematic.
Yeah. Okay. If there are no further questions, then to say thanks again to Johnny for an excellent overview of a very important topic in the immunology of COVID-19. And thanks once again to Hannah for bringing us right up to date with the evolution of the epidemic and all of that data from the Western Cape over the last 2 months. And we will be having our next COVID-19 ECHO webinar in 1 month from now. So back to you, Mark and Wendy.
Thanks everybody for attending again. Had really quite a very good participation, almost 150 individuals, still showing a lot of interest. So I think we'll continue to see you monthly for the next few months. Thanks.
Thank you, everyone.
Thanks.